Good morning, good afternoon, and good evening, everyone. I'm Ron Vale from Janelia Research Campus, and I'd like to welcome everyone back to Life Science Across the Globe. We're now in our third round of talks and very pleased to welcome back our sister institute, Cold Spring Harbor Laboratory. Cold Spring Harbor is a very interesting institute that does amazing science, but also has very innovative scientific programs as well. And in the past two rounds, we heard an interesting combination of science and science culture talks from Cold Spring Harbor, and we can look forward to the same today. So as one announcement, the clocks in the United States are moving back by 1 hour next weekend. So for next week's Life Science Across the Globe talk, we will start 30 minutes earlier. So that will be 8:30 in the United States. And that will make the time also a little bit more manageable for our colleagues in China. So right now it's a great pleasure to introduce Limor Joshua Tor from Cold Spring Harbor Laboratory. Limor is an acclaimed structural biologist and also a member of the National Academy of Sciences. And she will introduce today's speakers.
Thank you, Ron. Good morning, good afternoon, and good evening from Cold Spring Harbor Laboratory. It's so exciting to speak with you all over the world, wherever you are. Well, somehow I feel like it's the Eurovision Song Contest. It's a real treat for me to introduce my two wonderful colleagues, Bo Lee and Richard Seaver. I will introduce both of them before they give their talks. So first, Bo. Bo got a start in Jining, China for his undergraduate degree, and then the Chinese Academy of Sciences in Beijing for his master's. He then moved to the University of British Columbia in Canada to study with Lynn Raymond, and there he started to get his hands on NMDA receptors, studying their function and developmental regulation. After that, he decided to move to Robert Malinow's lab here at Cold Spring Harbor Laboratory for his postdoc to study synaptic plasticity in the hippocampus and the lateral habenula. He moved with Robert when Robert moved to UCSD towards the end of his postdoc, but then we were really fortunate to bring Bo Lee back to Cold Spring Harbor as a full-fledged faculty member. He did incredibly well here and is doing incredibly well here and moved up the ranks to now full professor here at Cold Spring Harbor. Bo is trying to understand aspects of motivated behaviors like attention, motivation, learning and memory, and also synaptic and circuit dysfunctions that underlie disorders such as anxiety, depression, autism. So with that, Bo, please take it away.
Okay, I hope you can hear me. Let me share the screen first. First of all, I want to thank Limor for the nice introduction, and I also want to thank all the organizers for this wonderful series of seminars from which I have learned a lot. So my lab has been focused on studying how neural circuits in the brain controls behavior and how behaviors can influence the expression of genes and also the function of molecules, which in turn can influence the synaptic and circuit function that ultimately can have an impact on the behavior itself. So the type of behaviors that we are interested in belong to the category of motivational behavior, which can be motivated by either reward or punishment. These kind of behaviors constitute essentially a large part of our life. And it can be very important for survival. For example, in order to survive, the lion needs to hunt for food, which is a major reward. On the other hand, in order to survive, the deer need to be able to avoid being eaten by predators, which is the major threat or punishment. So a major feature of this motivational behavior is that animals, including humans, are able to predict the future outcomes based on the past experiences. For example, if we see a sign like this, we know that something dangerous might be around. But if we see a sign like this, we know that something delicious could be available. So we can study this kind of behavior in the laboratory using animals. For example, we can study the punishment avoidance behavior using a paradigm called active avoidance. So in this paradigm, a sound indicates that a food shop is going to be delivered. But the animal can run to the other side of this chamber in order to avoid being shocked. So after training, the animal is able to learn so that when they hear the sound, they will run into the other side of the chamber and prevent being shocked. So in this case, the sound is called the conditioned stimulus or CF, and the shock is called unconditioned stimulus or UF. We can also train the animal for reward-seeking behaviors. And in this case, a light tells the animal that the food is going to be available, and the animal needs to learn to press the lever in order to obtain food. So in this case, the light is the CF, and the food is the UF. So we're very interested in understanding how neural circuits in the brain enable animals, including humans, to perform reward-seeking and punishment avoidance behavior. Not only because that these functions are critical for normal life, but also because that dysregulation of these functions is a core feature of several major psychiatric disorders, including depression, anxiety disorder, and drug addiction, and also autism. So extensive studies have indicated that the basal ganglia play a very critical role in motivational behavior. The basal ganglia is a system, is a brain system that is very well conserved across species. That is, the basal ganglia in the mouse brain is very similar to that in the human brain in terms of its structure and function. So the basal ganglia has several major components. The striatum is the major input component of the basal ganglia. It contains two major cell types. The D1 neurons express type 1 dopamine receptor, and the D2 neurons express the type 2 dopamine receptor. So the D1 neurons give rise to the so-called direct pathway because it sends direct projections to the GPI, which is the major output nucleus of the basal ganglia. The D2 neurons, on the other hand, are connected with the GPI only indirectly through some intermediate structures. So this pathway is called the indirect pathway. So according to a classical view, the function of the direct pathway and the indirect pathway is very different. That is, the direct pathway facilitates movement and promotes reward, whereas the indirect pathway suppresses movement and promotes aversion. However, recent studies indicate that neurons in the direct pathway and the indirect pathway have a coordinated activity as opposed to antagonistic activity that could be predicted from the simple dichotomic view. This is in the function of regulation movement, but the dichotomic view of these two pathways in regulating reward or aversion related behavior has remained unchanged. So on top of this division between the direct pathway and the indirect pathway in the striatum, there is another kind of division, which is called the division between striatum and the matrix. So striatum is a specialized structure within the dorsal striatum, and that has, in the location that has very high levels of expression of the mu opioid receptor. Okay, and the surrounding area within the dorsal striatum is called the matrix. Essentially, this striatum, which is indicated in blue in here, like small islands in the ocean of the matrix within the dorsal striatum. So many studies suggest that the striatum has very unique functions. That is, they seem to be important for reward seeking, for motivation, and for reinforcement learning. And also neurons in the striatum are particularly affected in mood disorders such as anxiety and depression. However, the challenge is that it's very difficult to study neurons in the striatum because this structure is unique in shape and has fuzzy boundaries with the surrounding matrix, making it difficult to precisely target those neurons. So as a result, how exactly the striatum neurons participate in motivational behaviors remain largely unclear. So to address this issue, we decided to develop a genetic method to target these neurons. A previous study indicated that a gene called TSHR1 is highly expressed in some cells in the dorsal striatum in a location that look like the striatum. And this result has been replicated or confirmed by a more recent study using single cell RNA sequencing from neurons in the dorsal striatum. So we decided to make a knock-in animal, for us to target neurons in this striatum. So this animal expresses flippase under the control of the endogenous TSHR1 promoter. When we cross this animal onto the reporter line, you can see that neurons, the TSHR neurons, shown in red in here, are distributed very widely in the whole brain, including the dorsal striatum. So when we look into the dorsal striatum, you can see the TSHR neurons form clusters or patches that resemble the striatum. To verify this result, we use immunohistochemistry stained for mu opioid receptor, which is the indication of the striatum. You can see that those TSHR neurons are highly enriched within this mu opioid receptor enriched domains, and suggests that these neurons are really enriched in the striatum. Furthermore, we use in situ hybridization to determine what kind of dopamine receptors the TSHR cell express. We found that these TSHR neurons express high levels of D1 receptor, but they don't express D2 receptor. This suggests that the TSHR neuron belong to the direct pathway. So to verify that these neurons are really part of the direct pathway, we use a compound animal that harbors both the TSHR1 knock-in allele and also a D1 transgenic allele. So in this animal, we can simultaneously label both the D1 cell and the TSHR cell. So remember, the TSHR cell is a part of the D1 population. So for example, in this animal, we use a red virus to label the D1 population and use a green virus to label the TSHR1 population. And this is the overlay of the images. As you can see, both cell populations send projection to similar or the same set of downstream targets, including the GPE, GPI, and the FNR. And those are all the known targets of the direct pathway. And this result really confirmed that the TSHR neurons are part of the direct pathway. Okay, so the TSHR neurons look like direct pathway neuron, but do they behave like a direct pathway neuron? For example, do they promote reward if we activate these neurons? So in order to activate these neurons, we use a method called optogenetics. For this purpose, we use virus to express a molecule called channel rhodopsin into the TSHR neuron in the dorsal striatum. And this molecule will make this neuron to be sensitive to light. We also implanted the optical fiber on top of this infected neuron so that we can deliver light to excite those TSHR neurons. And then we treat this animal in a protocol called real-time place preference or avoidance. So basically, in a chamber, animal are moving around, but whenever they enter a certain location, for example, if they enter the left side of the chamber, that will trigger the laser to activate this T-shaped neuron. And then when they exit this left side, the laser stimulation will be stopped. You can see that this manipulation really caused some aversive response to the animal. That is, the animal tried to avoid entering the location where stimulation of the T-shaped neuron would occur. So we call this real-time place avoidance. So this is quite surprising to us because when we, if we do the same manipulation to the general D1 population of neuron, it will cause an opposite phenomenon, that is the real-time place preference. So to further verify that activation of this neuron really is aversive, we treated the animal in a different behavioral protocol. In this case, it's a choice task. So the animal has the option to freely drink water in from two ports, the left port and the right port. But drinking in one of the ports, for example, in the left port, will trigger the laser stimulation of the T-shaped neuron. Okay, you can see that after the animal learned this task, it really try to avoid the port where the laser stimulation will be triggered, and only drink water from the other side, the other port. This is just the behavior response of one of the example animal. So from these two experiments, we make this conclusion that activation of T-shaped neurons cause aversive response to the animal. This is quite surprising to us because the T-shaped cell are part of a direct pathway, and yet they promote a behavior that is completely opposite to a conventional direct pathway would do. So the question is why? So in order to understand why and how T-shaped neurons participate in behavior, we decided to image the activity of these neurons during behavior. So for this purpose, we use a virus to express a molecule called GCAM6, which is a calcium sensor in this T-shaped cell. And then we implanted a very small length called the grain length on top of this infected cell, so that we can use a microscope to image through the grain length the calcium responses of the T-shaped neuron as a indication of the activity. So this is an example showing how the T-shaped neuron look like when they express this molecule GCAM6. And this is an example movie showing the activity of the T-shaped neuron in behavioral animal in response to different stimuli. We treated the animal with two kind of stimuli. One is aversive, that is an air pump blowing into the face, making the animal to be annoyed. And the other kind of stimulus is water reward. So this animal are thirsty, so water is very rewarding. You can see here is the traces, the activity traces from individual neurons across time. When we analyze all the activities of these neurons, we found that many of these neurons are activated by the air pump shown in red in here. But only few of these neurons are excited by the water reward. So this is the summary data from all neurons we imaged from different animals. You can see a major fraction of these neurons are only excited by the air pump, but not excited by the water. And a small fraction excited by the water, but not by air pump. So this is the distribution of the activity of all the neurons that we imaged for individual neuron. And this is the average activity of all the neurons. So maybe it's simple to look at the average activity. So which indicates that there is a major response across all the neurons to the delivery of the air pump, but almost there is no response to the delivery of water. So this indicates that the T-shaped neuron are primarily activated by punishment, but not by reward. So this leads to the hypothesis that these neurons probably mainly participate in behaviors driven by punishment. To test this hypothesis, we image the activity of these neurons in a punishment-driven behavior. which is an active avoidance task. So this task contains two types of trials. In the punishment trials, a sound predicts that air puff will be blown to the face, but the animal can run during a decision window to prevent the delivery of the air puff. If the animal don't run, they will receive the air puff. So in the neutral trials, a different sound predicts that nothing gonna happen, so that the animal do not need to do anything. So this is the behavior of the animal. So they can learn this behavior because, as you see, in many of the trials, the animal run during this decision window and successfully prevented the delivery of the air puff. But those animals do make mistakes because in some of the trials, they fail to run during this decision window and therefore receive the air puff. So these traces represent the running behavior in success trials or failure trials. So the running in the success trial is triggered by the CF, and the running in the failure trial is triggered by the air puff delivery. So when we look at the responses of the T-shirt neuron in the different type of trials, we found that they can be classified into three major functional classes. So the failure cells are the cells that only show response in the failure trial, but they don't show response in other type of trials, such as success trials or neutral trial. And the success cells, they only show response during success trials, but they don't show response in the failure trial. And there is a third category. Those cells, they do not discriminate between the failure trial and the success trial that show response to both trial types. Therefore, we call it non-discriminatory or ND cells. So I want to mention that the responses of this neuron seem to coincide with the running activity of the animal. However, we think that these activities do not simply represent the running or movement of the animal. Because when we look at the response carefully, so the failure cell, for example, they only are responsive during the running in failure trials induced by air puff, but they don't respond during the running in the success trial, which is induced by the CF presentation. Similarly, the success cells, they only respond during the running in success trial, but they don't respond during the running in failure trial. So we think that what is happening is that the failure cell may represent the value of the punishment. Therefore, they could be important for evaluation of this aversive stimulus. Okay, so consistent with this interpretation, we found that these cells, they have a graded response to punishment of different amplitude. That is, their response is higher to a bigger punishment as compared to a smaller punishment. For the success cells, we think that they represent the value of the expected punishment. And this could be important for motivating or invigorating the running behavior in order to avoid the shock, the air puff. So consistent with this idea, we found that many of these cells, their activity correlate with the running velocity of the animal on a trial-by-trial basis, indicating that these cells may be important for invigorating the running. And the ND cells may be important for both the evaluation function and also the motivation function. Okay. To verify that these neurons are really required for punishment-based behavior, we try to inhibit these neurons using a method called chemogenetic inhibition. When the animal are learning a task called the go-no-go task. In this task, there are two kinds of trials. In the go trials, a sound predicts that a water reward is going to be available, but the animal needs to lick in this response window in order to get this water reward. In the no-go trials, a different sound predicts that air puff is going to be delivered, but if the animal withholds the licking, the air puff will not be delivered. So we found that when we inhibit these neurons, the animal really has an impairment in the performance in the go-no-go trial as compared to the control animal. So when we look at carefully, we found that this impairment is specifically due to the impairment in performance during the no-go trial. That is, the animal cannot prevent leaking in these trials, and therefore received a lot of air path. And the performance is not affected in the go trial, in which they successfully obtained the reward. So this indicates that teacher's neurons are specifically required for punishment-based learning, but not for reward-based learning. So to summarize, by studying neurons in the striatum, we identified a unique direct pathway derived from the teacher cell. We call this pathway an unconventional direct pathway. This pathway encodes punishment as well as the motivation to avoid punishment. And they can cause aversive responses and also drives a negative reinforcement. We also have results showing that activation of this pathway can suppress locomotion or movements. And these functions are quite different or completely opposite to the function of the conventional direct pathway. So one thing I want to mention is that this gene TSHRT1 is deleted in patients in depression, and also has been shown that the deletion of this gene in mice can lead to depression-like behavior. So this genetic lesion likely can have an impact on the development or function of TSHRT1 neuron in this non-conventional pathway. And maybe impairment of this pathway contribute to some of the behaviors related to disease. This is something interesting to study in the future. So with that, I want to thank the people who actually did the work. This project is in charge by this brilliant postdoc fellow in my lab, Xiong Xiao, and he is really the driving force for every aspect of this project, with help from many people in the lab, including the current lab members and also past lab members. I also want to thank collaborators within Coastman Harbor and also the collaborators outside of Coastman Harbor, as well as different funding agencies for their generous support. I want to thank you for your attention.
Thank you, Bo. That was a fantastic talk, a really great story. And we do have a bunch of questions, so I'm going to just jump right in. The first is from Lou Sheffer, and I think it's a good question to get us all started. Why did you target the TSHRT gene and not the opioid receptor directly?
So the TSHRT gene motivated us because it's a transcriptional factor that may regulate many functions within the neuron. And also, as I mentioned just in the last slides, the human genetic study linked this gene to some mental disorder, and also animal studies show that this gene can also cause some depression-like behavior. So my lab is really interested in this kind of behavior. Therefore, we started with this, but I'm generally interested in studying the striatum. The other cell types within the striatum is also interest to us. We are currently working on different cell types in the striatum. It's a great question.
So moving on, do the TSHRT cells only express D1, or is it possible that they also express some levels of D2, which may be the reason why you're seeing these effects?
It's a great question. This is really a worry in the beginning that, so about 10% of the cell, the TSHRT cell also express D2. So one would worry that maybe the D2 cell function dominates in the behavior. But we do have some other result that make us believe this is not the case. So for example, in this experiment here on the right side, we stimulated the TSHRT cell, the fiber terminals in the GPI, and that will only affect those in the direct path, this bona fide direct pathway neuron, and we still see a worsening response. Okay, consistent with that the general population of these TSHRT cells is really different from normal D1 cell. And also in this experiment, we use fiber photometry to image only the tissue cell that send projection to GPI. Again, this is the direct pathway, the bound-of-fun direct pathway. Again, we found that they primarily respond to punishment but not reward. So we have some other data using this intersectional strategy to only target the D1 cell but not D2 cell within the tissue population, and we have the similar result. So we don't think our behavior or imaging result is contaminated by the D2 cell.
Great, thank you. So let's move on to a question from Junshul Park. Junshul, if you're there, can you unmute yourself and ask your question?
Yeah, so I was wondering if you have run a perturbation experiment with the active avoidance task, whether the success tissue cells were necessary for the active avoidance behavior.
Yeah, this is a great question. So we are doing it right now. We don't have the answer yet. But indeed, this is a good question to ask, because in the go-no-go task, we inhibited during the acquisition phase, which is the learning phase, and that impaired the learning. But after learning, we also did inhibition. There is no effect on the expression of the behavior. That is, the animal can still withhold leaking during the no-go trial. But we think maybe the tissue cell is really required for active avoidance. In the go-no-go task, the withhold leaking is a more passive response. The animal really don't need to make any effort to do that. But in the active avoidance task is a good paradigm to test. This currently we are testing that.
All right, so let's move on to a question from an audience member, Ritika Sood. Ritika, if you're there, you want to unmute yourself and ask your question?
Okay, hi, thanks.
Sure.
Yes, my question is, so you have different, in your experimental setting, you said there is different sound for the different groups. And so I was wondering, were there any animals that were common across these two groups?
I didn't quite understand the question. Can you?
So for example, in the reward group and the punishment group, there is different sounds that indicate whether there is going to be different. You said there is different sounds, right? For reward group, there is one sound plays, and then whatever the air, for example, the air in the animal's face or not, and then there is in the animals in the second group, you have a different sound that plays.
Yeah, so I think we use different paradigm. I think your question is the different paradigm, whether it's a...
Yeah, I'm wondering, is there a significance of using the different sounds for the two groups?
The different sound for the two groups. We normally counterbalance the kind of sound we use to make sure that it's not specific to the frequency of the sound. The major result is the same. Doesn't really matter which sound you use. What matters is whether you pair the sound with a reward or punishment. That is important. But the frequency or quality nature of the sound is not critical. Does that answer your question?
So a similar question is, would you, when you're talking about the specific nature of the reward or the aversive stimulus, does the nature of the stimulus have an effect, whether it's, different sounds or different aversive stimuli? Do they have, would you expect different results, or is it just any aversive or reward stimuli?
Yeah, it's a great question. We tried to image those neurons in response to different aversive stimuli. For example, the air puff and also the shock, tail shock. So the conclusion is the same. The difference that because the tail shock is more potent and it's more aversive, so we have more neurons responsive to the shock than to the air puff. And also for individual neurons, the response to shock is larger than to the air puff. But the conclusion is the same. They mainly responsive to aversive stimuli, but they don't respond to any kind of rewarding stimuli. If we try sucrose, the response is similar to just water. Minimal neuron respond to that.
So, Bo, I have another question here. Do you expect that there is any connectivity or some sort of interaction between the T-shirt neurons and the PDYN plus neurons, or are they just parallel pathways?
Yeah, this is a great question. So this is a diagram actually. So the question is in the striatum, there are two kind of neurons, the T-shirt one neuron and the P-alpha neuron, whether they are connected or not. So we don't know for sure because we didn't do the exact experiment. But according to previous study, they look at the connectivity in general within the dorsal striatum. The connectivity is kind of sparse. I think it's less than 10%. But doesn't mean that this connection is not important. I think there could be some interaction, lateral interaction between these two neurons. Additional interaction could be within the downstream targets. That is, although this cell grossly project to the same target, that is the GPI, but it still can innervate different type of neuron in here, therefore causing different activity in the output of downstream targets, and therefore leading to different behavior results. So that's the current hypothesis we have. I think it's mainly the connectivity within the downstream targets could be the key into determining the different function.
And let's close with this question from the audience, from Rashmi Banerjee. Rashmi wants to know, how does the T-shirt gene actually play a role in the behavior influenced by reward?
So we think that this gene, because it's a transcriptional regulator, could impair, let's say, a genetic lesion of this gene could impair the development of those neurons in terms of their migration and their wiring with their input and output. That could be one way to influence the circuit function and therefore behavior. Another way is that it may influence plasticity, let's say, in adult animal even. If this the transcription factor is required for synaptic plasticity, then it will be required for learning, especially for the punishment-based learning. So how exactly this gene regulates the function to link the gene to the behavior, I think we have a lot of work to do.
Excellent. Well, thank you, Bo. I think we're going to end it here so that we have time for the next talk. So I appreciate your presentation. It was fantastic. And now we're going to move on to Richard Seaver.